right, let's start with a sound check. Can you hear me in the back row? Am I loud enough in the back row? Yeah? Am I loud enough in the back row? Excellent. Do people have any questions about the upcoming test before we get into the topic at hand? Any other remaining questions? I'll be announcing shortly through this that there is the review session tonight that Susan will be conducting, which for some reason there's no icons for. It thinks so hard. Poor little computer. Okay. Here we go. Just a reminder that there is a open. All right, let's try that. Review session tonight here, 6 p.m. So you don't have to find a new room. There are a variety of resources on the web, on Icon, for study aids. And there will be, all right, let's try getting this open. And I have extended my office hours, but they're in the afternoon tomorrow. I am called up to Cedar Rapids tomorrow morning, so it'll be in the afternoon. Any other general questions? Let's go back. Meter options. Arrow options. All right. Any other questions before we return to the citric acid cycle? Yes. Thank you for reminding me. See, I need to be reminded of some things. Is there anything else I can do to make things work better? Okay. Fortunately, the people on the video don't know that there was a change in light. They just get the screen image that I have, which is why I don't notice the lights. All right. So just to sort of remind ourselves of where we are, after glycolysis, we ended up with 2 molecules of pyruvate, a 3-carbon compound. To get the rest of the energy out of it, we had to make 2 acetyl-CoA molecules through pyruvate dehydrogenase. And then feeding that into the citric acid cycle through a series of oxidation reduction reactions, we produced a lot of NADH, a couple of molecules of Q, and a couple of molecules of GTP. That's for a full glucose. One turn would produce 3, 1, and 1. And then this is where the CO2 come off. So the 2 carbons are fed in at the level of acetyl-CoA, 2 carbon dioxides come off. As a catabolic pathway, it has completely burned up the carbons that were put in the form of acetyl-CoA. So that's the citric acid cycle, thinking of it as a catabolic pathway. We talked a little bit about its regulation, the fact that since it's so energy intensive, the 3 main control points are going to be controlled by the abundance of NADH, which is the hallmark of abundant energy in the mitochondria. And a few positive feedback regulators here that are ADP certainly makes sense. And some feedback inhibition, so succinyl-CoA can inhibit its own enzyme, citrate can inhibit its own production also. We talked a little bit about the evolution of the citric acid cycle, that it looks at first glance to be something that is perfect and there's no place to begin it, but in fact in blue-green algae we can see what it was before it was a cycle. There's an oxidative branch down to alpha-ketoglutarate. It's good at burning carbons. There's a reductive branch down to succinate that's good at providing intermediates for other compounds. And the missing hole here probably could be filled fairly easily by small changes in pyruvate dehydrogenase, which has many, many homologies to how alpha-ketoglutarate dehydrogenase works. Both of them are going to decarboxylate, both of them are going to make a CoA molecule, and so on. When people were first studying the citric acid cycle, this was the pathway that was initially proposed. And you can see it has the same sequence of compounds, but it's going around in the opposite direction, and it's acting to fix carbon dioxide to generate a molecule of pyruvate ultimately. What is wrong with that picture? You can talk to each other if that'll help at all. Any thoughts of what's implausible about this picture? Anyone want to venture a possible guess? Yes. There's a problem, you think, on the right side here. What do you think the problem is in there? Mm-hmm. Right. So in other words, for this pathway to work, you'd have to get this reaction, that reaction to go in the backwards reaction. This one also is usually a forward reaction. And we've seen some cases where carbon dioxide is fixed into new molecules. For instance, when we were making gluconeogenesis work, we had to invest GTP to get a carbon dioxide back into a molecule. A pathway with the carbon dioxide spontaneously popping back in was not very plausible. And the reason they could get confused this way is because in the test tube, you can set it up with 1,000-fold excesses of product. And when you can have a 1,000-fold excess, you can drive that thing backward, but it will never go backward in the cell. So it was not ever a full cycle functioning for reduction. Now, we promised at the beginning that the citric acid cycle would be an example of an amphibolic cycle. And so we've looked at how it's catabolic, that if you feed in carbon backbones in the form of acetyl-CoA, you can oxidize those to CO2. But there are a lot of the citric acid cycle intermediates that can be used to make other compounds. And it's because those intermediates can be pulled out of the cycle and used to make other compounds that we call it also an anabolic cycle. And since it can be either catabolic or anabolic, it's amphibolic. And there are times when it's both at once, just to make life more interesting. To give you an example of some of the things that get pulled out of the cycle, succinyl-CoA can get pulled out to make heme. Alpha-ketoglutarate can be pulled out to make glutamate. So now you know where the glutarate comes from. It's a kissing cousin of glutamate. It's the just one group away from being an amino acid. Once you've made glutamate, you can make glutamine. You can also withdraw backbones for glutamine, for arginine, for proline. Glutamine itself can be used in the biosynthesis of the bases for nucleic acids. Now this is an interesting reaction, and it's a useful reaction for a number of reasons. So here we have an alpha-ketoglutarate. This would be an intermediate in the Krebs cycle or the citric acid cycle, thinking of it catabolically. But if you have ammonia around, this enzyme can combine that ammonia with the alpha-ketoglutarate to produce a glutamate. So where there is a carbonyl carbon here, there's now a primary amine. So the alpha carbon here is the receiver to become an amino acid. That is a case where you have to reduce that carbon. And so in order to reduce it, you're investing some NADH, and it comes back out as NAD+. Now, this can be thought of as a synthetic reaction. That is, you're making an amino acid. It's unusual for there to be an NADH without the P used for an anabolic reaction. But you notice that this is a reversible arrow, and it's a reaction that can be used to make glutamate or can be used to get rid of ammonia, depending on rid of amino acids, depending on what your current metabolic need is. And I think that's why this is an exception to the usual rule. It's because it is a reaction that is itself amphibolic. And if you think about it, a number of the steps in the glucose glycolysis or gluconeogenesis pathway are amphibolic because they can either be glycolysis or they can be gluconeogenesis. So this gives you some of the destinations these things are heading off to. The citrate backbone, you think about citrate at 6 carbons, it's probably not too surprising that that ends up being precursor for carbon-rich compounds like fatty acids, cholesterol. We've just seen how alpha-ketoglutarate can become glutamate, and From there, glutamine, and thus a precursor for amino acids and nucleotides. We've talked about succinyl-CoA. That's that 4-carbon backbone. And you remember that heme has that kind of flowery look to it. Malate can get pulled off to make pyruvate. Oxaloacetate can get pulled off for gluconeogenesis, and so on. So altogether, the intermediates from the citric acid cycle can go to all 3 major groups. They can end up in fatty acids, they can end up in proteins, they can end up in nucleic acids, and they can end up in carbohydrates, all 4 groups. So all 4 major groups they can end up in. Part of what makes the citric acid cycle work, but also makes it tricky, is of the control over where it is. And although we haven't emphasized this so far, what we've been talking about so far has been happening in the matrix of the mitochondrion, in the interior goo that's all the way at the inside of the mitochondrion. And for that matrix to be able to get at things, it has to be able to move carbon backbones around in ways that let them be useful. So for instance, if you want to withdraw citrate to make fatty acids, the citrate is in the matrix, fatty acid synthesis is in the cytoplasm. In that case, you can make a citrate molecule, and a transporter will shuttle the citrate to the cytoplasm. An enzyme in the cytoplasm called ATP citrate lyase can make an acetyl-CoA and an oxaloacetate out of it. So this is the reverse reaction of that one. We're getting out an acetyl-CoA and an oxaloacetate, but it cost us an ATP, and it's a cytoplasmic enzyme. That acetyl-CoA now can go off for lipid synthesis. The oxaloacetate, on the other hand, is outside in the cytoplasm, and you'd like to get it back in. There's a cytoplasmic malate dehydrogenase. In this case, it's driving in the other direction than in the mitochondria. An enzyme called malic acid can pop off that CO2 and give you a pyruvate to come back in. And so this cycle of carrying carbon backbones out is driven through ATP investment here and the decarboxylation steps. And of course, there's GTP investment there. Take another look at the malic enzyme here, and this is another decarboxylation step that ends up with a reduced adenine nucleotide. In this case, it's NADPH, and that reflects the fact that this is going to be principally active in providing carbon backbones during a more synthetic phase in the cell. Now, it should occur to you that if we draw carbon backbones out of the citric acid cycle in order to make other things, at some point the citric acid cycle will be empty. And so there have to be some other ways of putting things into the citric acid cycle. And it's true. As a family of reactions, they are called anaplerotic, filling up reactions. That's just Greek for filling up. By controlling the anaplerotic reactions, the cell can control how fast the citric acid cycle itself is spinning. So we just saw that the pyruvate carboxylase can give us oxaloacetate. We saw that initially in the context of gluconeogenesis, but that can also be an anaplerotic reaction to make more citric acid cycle intermediates. If there are fatty acids that have an odd number of carbons, they can add fresh backbones in at succinyl-CoA. Amino acids can put in their backbones in a number of places. As you might expect, alpha-ketoglutarate is one of the big spots since it's so closely related to an amino acid, but fumarate is also another one that can donate carbon backbones to refill the citric acid cycle. So it's worth thinking about this relationship between amino acids and the citric acid cycle. We've seen them come out as a product of the citric acid cycle, But they can also be used to refill the citric acid cycle, and they can also be sort of muddled with. They can be rearranged a bit through the citric acid cycle to make better precursors to make sugar or fat. So if you have a lot of excess energy and spare protein, the spare protein can be stored not in the form of protein. You can only store protein in muscles, but in the form of fat or sugar. And here are a couple of the kinds of reactions between amino acids and other metabolites that can do that. So for instance, as a class, we talk about transamination reactions where an alpha amino group like this can be exchanged for a carbonyl oxygen. And so this carbon turns from having an amino group into having a carbonyl oxygen. That carbon picked it up, and now it has an amino group and is an amino acid. In the particular ones we're talking about in this example, we're starting with an aspartate and a pyruvate. We're ending up with an oxaloacetate and an alanine. So we end up with an intermediate here for the citric acid cycle itself. Another example of the kind of transferases that can go on. Here we have a pyruvate. We might have just made it up there, in fact. And if the pyruvate undergoes the aminotransferase reaction with a glutamate, you can end up again with an alanine and now with an alpha-ketoglutarate. So aspartate can enter the citric acid cycle as oxaloacetate. Glutamate can enter as an alpha-ketoglutarate. And you can remember this because this is a 4-carbon fragment making the 4-carbon oxaloacetate. This is a 5-carbon amino acid making the 5-carbon alpha-ketoglutarate. It's a frustrating but true fact that we cannot make sugar out of fat. We can make sugar if we have both protein and fat, but we can't make it just out of fat. And the reason for that is a little subtle. So if you bear with me, I think it'll come clear. Plants can make fat out of sugar out of fat. And this is the trick that they use to do it. They start from acetyl-CoA, and we're going to see, we've mentioned earlier in chapter 9, but we're going to see again, that acetyl-CoA is the common breakdown product from fatty acids. So you can feed acetyl-CoA in what starts looking like a citric acid cycle to make a citrate and an isocitrate. But now, instead of losing a carbon dioxide, we take off a 2-carbon fragment, a glyoxylate. So instead of blowing off 1 carbon, we take off 2 carbons at once through isocitrate lyase. And now we go straight to succinate, do not pass alpha-ketoglutarate, do not collect $100. And at succinate, we pick up with the rest of the reactions of the citric acid cycle, which go in the normal way, and you can go around again and again and do this as often as you like. Well, what good does that do us? It gives us a glyoxylate fragment, so it's a 2-carbon fragment. And if we take a second acetyl-CoA, which we'd have on hand if we're breaking down fats, we can make a molecule of malate out of it. And now we have a 4-carbon backbone. And now we have oxaloacetate. And we know that we can take oxaloacetate and do gluconeogenesis with it. So we're seeing a number of different ways that we can make oxaloacetate in the cell. What the glyoxylate cycle does for plants is it completely bypasses the decarboxylation reactions. Instead of blowing off 2 molecules of carbon dioxide the way our citric acid cycle does, this cycle plucks out a 2-carbon fragment and then picks up, where it can, which is at succinate. In our bodies, since we break down fat to acetyl-CoA, and we don't have the glyoxylate cycle, all we can do with fat is burn it for energy unless we have a source of carbon backbones. And that source of carbon backbones could be, for instance, from amino acids. But fat alone can't give us carbon backbones to make sugar out of. Any questions about that before we move on? This is a very important reaction for plants because, as I'm sure you know, being many of you Iowans, many seeds have oil in them. And the oil then is not merely the energy for the process of sprouting, but it can actually provide it with some of the sugars they need as well. Now, this was the originally proposed citric acid cycle, or we can call it the Krebs cycle since we're talking about Krebs' version of it. And in that pathway, he assumed that pyruvate could react directly with oxaloacetate. There's no intermediate step there for pyruvate dehydrogenase. And he imagined that the carbon dioxide coming off was what drove the reaction forward to citrate directly. And in a reaction like this, it appeared to him that it ought to be the carbons that entered in pyruvate ought to come out symmetrically on both sides. And yet, it didn't work that way. Although it looked as though it ought to come out symmetrically, it didn't. And this is why they thought it should be symmetrical. Here you're starting with your oxaloacetate that you're going to make into a citrate molecule. So you've added on an acetyl-CoA to it back here on its fanny. And there's the carbon at the citrate. And now if that molecule goes to isocitrate, goes to aconitate, in this formation, one could imagine that it could have formed that aconitate in either side. In other words, it could have had the radioactive carbon or the labeled carbon at the top or at the bottom. And if the aconitate had it at the top, then the isocitrate would have it at that end, and the alpha-ketoglutarate would have it down on its tail. If it had made it this way, then you'd expect to see it on that end of the isocitrate and on that end of the alpha-ketoglutarate. So on the face of it, it ought to have scrambled the carbons at the stage of citrate, and yet, when they actually looked at alpha-ketoglutarate, only one of the two carbons, in fact, were labeled. So something was going on to restrict the formation of the molecule from simply scrambling or being random enantiomers. So they then tried to modify their first pathway. You can see here we still have this pyruvate coming in directly. And then they wanted to suggest that it came in as a conatate, and there was a side reaction making citrate. But this actually doesn't solve it either. What actually did solve it, and this was a major insight, was realizing that when a molecule like citrate, which has a mirror symmetry to it, right? If you think back to the structure of citrate, it has mirror symmetry. That when the citrate bound to the surface of the enzyme, those groups were no longer equivalent. That there were some that were in contact with the enzyme and some that weren't. So these two green ones are in a mirror symmetrical relationship to each other, but the blue one and the red one can only contact the enzyme at the blue patch and the red patch. And so that forces one of the two green ones down. The other one can't go down. And so the surface of the enzyme is the structural basis for having that limitation. So once they get to succinate, however, we do start to see the carbons thoroughly scrambled. I've mentioned that biochemistry is nearly universal, but it's not utterly universal. I've already mentioned for you the fact that the blue-green algae don't have alpha-ketoglutarate. There's some other microorganisms that sort of are halfway there. They have an imperfect citric acid cycle, so to speak. And for those microorganisms, they go from alpha-ketoglutarate to a succinate semialdehyde, and then to succinate. And that reaction involves a decarboxylation to get from the 5-carbon alpha-ketoglutarate to the 4-carbon succinate. And this is the semialdehyde part of it. And when this is oxidized, you can reduce an NADH and come out with succinate. How does this version compare in energy yield to an authentic citric acid cycle? We can talk to try and figure that one out. So how does this compare in yielding energy to a normal citric acid cycle like we would have? You on the video, you can work on it at home. Any thoughts? Any brave souls willing to take a stab at it? Can I put you on the spot? You want to take a stab at it? No? No? Anybody willing to take her off the spot? All right, let's think this through then. Coming out of alpha-ketoglutarate in the ordinary citric acid cycle, you'd have alpha-ketoglutarate dehydrogenase, which would decarboxylate, like this reaction does, but it'd make an NADH, and it would make a succinyl-CoA. And then the succinyl-CoA would react to yield a succinate, but it would produce a GTP in the process. So we have the same number of NADHs, although they're in slightly different position. We're going to be one high energy bond short compared to the other pathway. So it's actually fairly close. Not a huge difference in efficiency, but you don't make the succinyl-CoA. So you have reduced the number of sources you have for making heme groups and things like that. If this was an anaerobic microorganism, they wouldn't mind not making as many heme groups because they wouldn't have as many heme groups. So if you were to predict, you might predict that this would be seen in an anaerobic microorganism. So if this is an inhibitor of the citric acid cycle, and we're showing the rate at which the cycle is going, and the variable on the bottom here is acetyl-CoA being made. So this is citrate synthase, for instance. What kind of an inhibitor has been added to this reaction? Sorry? So you're suggesting a competitive inhibitor. Would you like to give the logic for your answer? All right, so you're saying Km increases, and that's because the horizontal axis is going to be concentration, and the intersect has come over here, but since it's a double reciprocal plot, that's actually a larger number or a lower affinity. And then if we were to look here, the Vmax is unchanged, so quite right. I think that's the end of what we have on the citric acid cycle. So I will throw it open to any kind of further discussion of the citric acid cycle or to review material for the exam on Wednesday. So what would people prefer? Mm-hmm. I have mentioned that. It's only through Chapter 10 explicitly, but do bear in mind that there are things in Chapter 9 that are repeated in more detail in Chapter 10. So whatever depth they're in Chapter 9, that's a legitimate question. And if you've been paying attention for the last two lectures, you'll have refreshed that material along the way. Chapter 11, sorry. This is Chapter 11. Chapter 9 was the overview of metabolism, and it discussed the citric acid cycle in an overview kind of way. It discussed the electron carriers and oxidation reduction compounds and things like that. Other questions? Would anybody like me to go back? Yeah. NADPH is almost only in biosynthesis. It's very hard in biochemistry to say only. But if you see NADPH, you should think biosynthesis. That's the most likely thing. NADH, NADPH and NADH have almost exactly the same electron reduction potential. That is, they have almost exactly the same amount of reducing power. And so there's not a difference at that level. The P is at the other end of the molecule. If you look at the structure of NADP and NAD, the P is way off yonder. And all that does is allow enzymes to know which ones they're using. And if you think about it, what that means is that the cell has, in effect, two pools of reducing power that are coexisting without getting confused. There's the pool of NADPH and NADP+, and the pool of NADH and NAD+. And they can be in the same space without getting confused because the enzymes will use one or use the other. And so it allows the cytoplasm to do catabolic things like glycolysis for energy and anabolic things like making nucleotides and do that all in one cytoplasm. Does that help? Okay. Other questions? I can also open up any of the handouts from previous chapters if you want me to have those open to discuss. Yes? Explain the graph one more time? Certainly. This is what's called a Lineweaver-Burke plot. If you think of the simple direct plots for enzyme activity, they give you either a hyperbolic curve or a sigmoid curve. And on graphs like that, it's hard to estimate exactly what Vmax is. And if you can't know exactly what Vmax is, you can't know exactly what Km is, because Km is related to Vmax. It's the concentration of substrate that'll get you halfway to the Vmax. And Lineweaver and Burke came up with the idea that if you were to mathematically transform the Michaelis-Menten equation with double reciprocals, you would linearize the hyperbolic plots. And so this lower axis is 1 over the concentration of acetyl-CoA. And this axis is 1 over the velocity. So that's why we call it a double reciprocal plot. And it does have the good effect of making those lines linear. It has the drawback of making them reciprocals. And so the further up you go here, the bigger the smaller the number. The further out you go here, the smaller the number, because it's a reciprocal plot. Well, on a Lineweaver-Burke plot, the intersection with the vertical axis, the enzyme velocity, will be the Vmax, because this is the smallest velocity it has. And the intercept with the substrate concentration will be 1 over the Km. And so if the Km is moved this way, it's now a bigger number or a less affinity. Does that help clarify that graph? Now, it's a good way to transform hyperbolic data. You can't use this method for sigmoidal data because it won't get straight. A sigmoidal group of data is not going to get straight when you do this to it just because you put it through a reciprocal plot. So there are other ways of finding Vmax and Km for enzymes that have sigmoidal activity. The Y, sorry, your question again? Yes, that's 1 over Vmax. So this is it's a double reciprocal plot, so you're going to see 1 over Vmax. You can deduce the Vmax from that. Mm-hmm. Does that clarify the relationship between a direct plot and a Lineweaver-Burke plot? Does anybody want me to clarify any points on that otherwise? Okay. Any other questions? Yes? I'm sorry. Is this graph more trouble than it's worth? The other graph is very intuitive. It's very, you get the sense of it right away. It is not very precise. And so you can't get a very good estimate of the Vmax. And without a good estimate of the Vmax, you can't get a very good estimate of the Km. So enzymologists who need precise numbers to make decisions about things like what order things enter the enzyme or the true affinity of things needed something more precise. To be completely honest with you now, there are computer packages that can in fact deconvolute things and give you extrapolations, because this is an extrapolation obviously. But for many years this was the fastest way for enzyme kineticists to get the kinetic parameters that they needed for their numbers. Other questions? Mhm. Mhm. Well, would people find it helpful to see the citric acid cycle one more time from an eagle's eye view to get that grand scheme sense of the pathway? So you don't just focus so much on the specific steps, but the grand view of it, or not. Mm-hmm. Okay. I will remain here until the end of the hour if people have questions.
Better computer for you?